I thought just a little preface here. The brain has all this complicated anatomy. It's been documented for decades and decades, for over 100 years. It's really messy. There are different terms that are used for the same things. There's different reported empirical results that conflict with one another. There are empirical results that are on the same topic, but because of the different techniques in animals and so on, they're talking about the same thing, but there's no overlap between them. And I've said this many times, if you want to know anything about the brain, you have to read at least 7 papers before you start sort of getting a gist of, okay, what's really happening here? Because an individual paper can be very misleading, not wrong, not incorrect, but just not using the same language, the same things, or whatever. You just get different answers. It's also true that the anatomy itself is not like a circuit diagram you can look at and deduce what is going on. You just can't do that. It's just too many unknowns, too much complication. It's not possible. So give up on that idea that you could look at it and go, oh, I can deduce what's going on. There's a few simple circuits and biological neural circuits that people have been able to do that with, but not anything close to neocortex. It's just crazy. And there's so many, we're still discovering how the neocortex represents information and so on. So you can't do that. So what good is this studying anatomy? The good of studying anatomy is that it's a classic science problem. You have top-down theoretical constraints, which you say, okay, the theory and deduction says me this has to happen. And then you can look at the anatomy and say, well, can that thing that has to happen match up here at all? And how would it match up? And maybe there's only one way to do it. Maybe there's multiple ways to do it, but at least it's this interaction back and forth. And then you say, oh, well, I wish I knew more about the anatomy because that may prove this theory. Then you dig and you find out more about the anatomy and physiology, and maybe it disproves it, proves it. So my point is, on its own, the cortical anatomy and physiology, with brain anatomy and physiology isn't, it can't do anything with it on its own. You have to match it with deductive and top-down theoretical constraints. So that's the process that we've gone through. Any questions about that before I dive into any further? All right, there's one axiom that we follow, or I follow, and we all follow here. An axiom is just something you just take as a truth, right? It's a ground-based, you just have to accept this. And this is Mountcastle's proposal. This is the cortical column hypothesis, which is that the cortex, which is this big sheet of neural tissue, is divided up into these columns, and they're all basically doing the same thing. And it means it's not purely made up. There's a lot of evidence for it, but it's the one thing I don't question as we're going through our work. I never go back and say, I wonder if Mountcastle was wrong about this, because we could come up with some, the consciousness circuit over here or something like that. I just don't go there. There's the one sort of axiom that we stick with. And so it's underlying everything. Hopefully it's right, because if it's not, then we're all screwed. But so far, there's a lot of evidence for it. And I think it's really safe.
One thing maybe on this is, so like in terms of how you define the extent of a column.
Right.
Because I think sometimes there's more like a histological point of view, and then Numenta kind of has like a. Well,
so part of Mountcastle's proposal, it talks about what is the extent of a column. So this is your neocortex. It's about 3 millimeters in thickness, and it's a big sheet, right? And different parts do different things. And when they first observed it under a microscope back to Cajal, they saw that the predominant architectural feature was layers. So and you've seen the picture, hopefully, of the picture that Cajal drew, but you could see there's different cell densities and different cell types, and it kind of look like these layers of cells, and these just extend across the cortex continuously like this. So, and they came up with 6 layers. That is incorrect. There aren't 6 layers. There are different cell types and different densities, and they just made up the number 6. And so, for example, today, generally, some layers are grouped together. Layers 2 and layers 3 are often just referred to as layer 2, 3. Some people don't make a distinction. There are papers that talk about layer 2A and 2B, and if I write correct, and layer 3A and 3B. Well, who's right? I don't know. Depends on how you judge it, right? You can look at cells and say, oh, in between 2 and 3, their cell types are a little bit smaller. They make slightly different projections. You say, okay, I think those are really separate, even though sometimes people don't talk about it separately. Layer 5, it's often like there's 2 types of cells in layer 5. But now there's actually a 3rd type that people are arguing about that connects to the striatum or something. And then layer 6 has got like half a dozen different types of cells in it, at least. So these layers, visually, you can see them. Maybe you could count 6, but they're just placeholders that help you kind of locate cell types relative to each other. But we often fall into the habit of saying, oh, layer 6A connects to here. Well, even in layer 6A, there's more than one cell type. So we have to be really kind of conscious about it in the back of your mind. The next thing is that these cells almost all, the next thing is that the general rule is that if you look at information that comes into the cortex, information flows up and down vertically. You see a lot more vertical connections than horizontal connections. So there's clearly a lot of information going back and forth, up and down. And then there are connections in some layers that go small distances or long distances. Sometimes they exit and go out and back in again. So there's a very heavy connectivity vertically oriented, and then a sparser connectivity, but still a lot going elsewhere. So this is the first suggestion that there might be columns, because it seems like the information is first and foremost processed in a sort of vertical fashion. And then Mountcastle made the argument that that if you look at the cortex and you look at like what you might call a column, that what distinguishes these between them? There's no visual demarcation between column A and column B. You can't see a line between them. It just looks like layers. But he made the argument that a column is defined by where it's getting its input from. So if it was like on your hand, here's a famous picture where this is on the hand of a monkey, the skin of the monkey, and these columns are getting input from a patch of the skin, but it's not continuous. That is, all the cells within a column are getting input from this patch here. But if you skip over to the next column, just move a little bit further, they're all getting input from here. And you skip over a little further, then they're all getting input from here. And of course, sometimes in the cortex, you go from one region to another, or one modality to the other. So this might be a somatosensory column, this might be a visual column, and they're getting input from a completely different sensory organ, a completely different part of the
Are these sensory patches non-overlapping, or do they overlap?
The sensory patches do overlap a bit. If they didn't but not a lot, not completely. And this is his argument. He gave evidence for this. But the most important thing is it's not about the overlap. It's the fact that all these cells from here to here all are driven by the same area. And as soon as you jump a little bit, they're all driven by a different set of things. It's not a continuous thing. It's a sudden jump. So he said that is sort of his proof that these are separate processing units. This thing here processes all the information from this patch. This comes it's only getting information from this patch, and so on. And the same thing, it happens in vision and touch and some in auditory, and he gave evidence for that and all those. So this is his proposal about columns. And then he said, well, since they all look identical, pretty much, then they're all doing the same thing. They're just processing a different part of the world. And again, remember, I could have a vision column right here next to a somatosensory column, or I can have a V1 column bordering. They're not intermixed, but they're points in the cortex where there are boundaries between these regions. And if you just jump over a little bit over the hoop here, all of a sudden you're in a different modality. In fact, it was, what's his name, who proposed that synesthesia is caused by cells incorrectly making connections across modalities like that. What's his name? Who did that? The famous, don't come to me. Anyway, so that was again evidence like, yeah, there's these, there's these hard boundaries sometimes, but then even here within a single modality, there are these boundaries between one. So then you can just think like, okay, a column it's just looking at a patch, it's getting the input from that patch, and that's all it does. And then it processes it. There is another confusing element called minicolumns. So if you're not familiar with that, a minicolumn is, so right, again, columns you can't see them in the cortex, right? The only example that's different is the rat barrel cortex. So rats have a column for each whisker, and that you can actually see those. Yeah, but generally you can't see column. And but there is something you can see called the mini column. And now I'll just start like this. And the mini column is much, much smaller, where a regular column can be anywhere between like a third of a millimeter and a millimeter in dimension. Mini columns are much, much smaller than that, maybe like 30 to 60 microns or something like that. And it's typically reported, each mini column is like a little stack of neurons that go across all the layers. So the general idea is that all the different types of cells that exist in a column exist in a mini column. And so a typical number, you'll see different numbers, but 120 cells is a typical number you'll see for a mini column. And they come about when you're in utero and you're developing your brain. The way the brain develops, the cortex develops, there's a sheet of neural tissue, just a single layer, and then the cells start replicating, and they move vertically. Well, they move in this direction. And so essentially a single progenitor cell ends up creating 120 cells, and they grow up vertically like that. So that's how they come about. So the mini column is a real thing because that's how the cortex develops, and you can often see them under a microscope. There's these great pictures. You see these little skinny little cells, and there's a little gap in other skinny little cells. They're not always visible. So in some animals, like in rats and mice, it's hard to see them. Or they don't look like they exist. And people say, well, they don't really exist. They say, oh, they were there when your brain grew, but they're not there now. And other people say, well, even if they are there, they're not functional. Like, what do they do? Like, maybe it doesn't matter. It's just the way they grow. Malk has argued that this actually was the most important replicable unit of computation, but he had no hypothesis what it was. He said that is the unit of replication in the brain, the minicom. And he said if you take a bunch of minicoms, several hundred minicoms, you put them together, they form a column, and they work together. So this is the two levels of organization that you see in the cortex. I have a theory about minicoms. I've never really shared it completely with everybody. We can get into that later. But I'm developing a partial understanding of how would I think about what does a minicolumn do as an independent unit. Obviously, you can't do too much, right? Because it's just 120 cells, and obviously you can't do. They have to work together as a group. But can you understand what they do? I think you can. So, so that tells you, if you said, oh, there are 6 layers of cells, and you might say, well, there's 20 layers per cell, 20 cells per layer, or something like that. But we've already seen that that's not really true that there's 6 layers.
In terms of like the lateral extent, so I think sometimes when I've read about a column, it suggested it's like around 10,000 neurons. But my understanding is
they're much bigger than that.
Yeah, it's more like 100,000 at least. But I thought that was arrived at in the mental stuff through some other logic.
If you look at the cats, the hypercolumns that Hubel and Wiesel talked about in cats, those are about a millimeter square in area. And basically, just do the math. In a square millimeter, you'll have about 100,000 neurons. Okay. Now that's arguably the hypercolumn in a cat. I haven't really gotten definitive answers on it. Is that a column or is that multiple columns? It's kind of debatable. But at least at the high end, you might have a column which is a square millimeter in area, 100,000 cells. If your column was smaller than that, let's say it was 300 microns by 300 microns, that would be like a tenth of that. There'd be like maybe 10,000 cells like that. The numbers generally work out well. That is, you cross correlate them, but they don't always fit properly. There's also evidence people have argued that some of the cells in the cortex do not start in minicom. Some of the inhibitory cells generate through a separate process. So there's more cells than in minicom. It's complicated.
That's not my question. Like the minicoms make up all the neurons in a column, and you just said
yes, but maybe no. No one really knows. Different people argue different things, but the general answer is yes. The general answer, there are other cells that are not. So if you look at the cortex, about 75% of the cells are excitatory cells. These are cells that you input, they have a positive, they affect other cells in a positive way. They make other cells fire. Then roughly 20 to 25% are inhibitory cells, which make other cells less likely to follow. Definitely all the excitatory cells, I believe, are in minicolumns. Some of the inhibitory cells are weird. There's these ones in layer 1, which hardly anyone ever talks about, that are really weird and shalong things, and they don't really fit one per minicolumn, that kind of thing. So who knows? One of the rules I have about neuroscience is that for every rule, there are exceptions, every single one. And so I can say all the cells are there, and then someone says, no, they're not. I can say all these are all excitatory cells, and say, oh no, here's one. Never ending about that. Okay, so that's the general layout of this common algorithm, divided into columns, each one getting a patch an input from someplace, and the next column processing input from someplace else. And then that being divided into minicolumns, which our theories generally don't talk about. But if you look at the temporal memory algorithm, it's really relying on minicolumns. So we do take advantage of it. But in our Monty today, I don't think we do anything with minicolumns. We just take the concept of it, right? So this is any questions about this now?
So the proximity in the sensor patches, is that mapped into the proximity between columns?
Right. They're generally except when you get to borders, right? If you switch from one modality or one region to another, there's a discontinuity. Yeah.
So cortical column computational unit, mini columns computational unit. In the cyber architecture, different areas, different Brodmann areas will have different looking layer distribution.
No, well, the same layers. The differences would be that you mentioned Brodmann. Brodmann's a guy who tried to define regions by looking at the visual differences between parts of the cortex. So the typical variation you see is the number of cells in each layer can vary. One region might have more layer 3 cells, one region have more layer 5 cells. The cell types may vary in size. So like what people classically call the motor cortex, which have neurons which project to your spine, those cells are really big because they have to project a long way. But the same cells exist in other regions, but they're smaller, even though the same cells, they're just smaller cells because they don't project very far. Maybe they only project to another part of the brain. Those are all motor output cells. They're all layer 5A cells. But some will look at the motor, quote, motor cortex, which is really somatosensory cortex, will say, oh, those cells are different, they're bigger. So we can define the motor region because it's got these big cells in it. But functionally, it's not really any different than regions that have smaller cells in the same place.
That was going to be my question. So that distribution, that doesn't mean anything that maybe for motor system, it had to optimize for different.
No, it does mean it could be, and it almost certainly is.
Okay, good.
So there are the classic big exceptions to this common algorithm. One is motor cortex because of these big cells, which people at the time didn't realize are the same as the little cells in other regions. And the other classic difference is the primary visual cortex, V1, which is we see is the first region that gets input from the retina, has some extra layers in it. In fact, a classic V1 in a primate has about twice as many cells per minicolumn. But it's only, and they call them extra layer 4 cells. So they have a few extra layers there. Now, here's the thing about that. So obviously that is an optimization on vision that works better. But many mammals don't have that. Dogs and cats do not have what they call a striate layer 4. They don't have those extra layers. So presumably this is something that primates have evolved to have somehow better vision, somehow different than the vision of dogs and cats. So, but dogs and cats still see. So how do we handle that? The way I handle that from a theorist point of view is I say, okay, clearly these are important, these extra layers, but they're not essential for the basic algorithm of the cortex. They don't exist in touch, they don't exist in hearing, they don't exist anywhere else in the cortex, only in V1, and not all animals with eyes that see have a striated cortex. So it's important, but it doesn't overthrow our axiom. It's like, and so the general idea I've always said is we can just focus on the core commonality across all modalities, because there seems to be the same basic arrangement of cell types in all modalities. And then the variations we see, whether it's an extra few layers or extra bigger cells or more of these cells and less of these cells, they're all tweaks that, and so we don't want to focus on those. We want to focus on the core algorithms first, what's going on common to all of these things. And then later we might say, even in our work, we're learning module, we might say, hey, you know what, we're going to tweak the learning module for LiDAR. Here's how we could do it. We can add a little extra twist here and put some more of these things here. And, you know, I don't know. We can do that, but let's not focus on that. So don't get hung up on that. Good question. Any other questions before I go on? All right. I know some of this, for some of you, this is all review. Some of it may not be. I'm now going to rate this if I can, or should I just turn it around and go on the other side? Sure. Does it turn around?
Maybe you can flip it.
Oh, I can flip it.
Second
Oh.
whiteboard over there.
We can flip it. Oh, I see. Oh, this is clever. And the wheels are locked. There you go. Okay, great. Thank you. So this is a classic example. If you were to go to a neuroscience and one-on-one class in any university, and you're talking about the brain, and they would say, here's the cortex, and here's a column, and they would say, okay, what's going on here? They would say the following. They say, okay, there's layer 4 is the input layer. So it comes from some, I'm going to draw it as if it was coming from the outside here, like, oh, like this, okay? Layer 4 is the input layer, because this is where the eye projects to, or the skin projects to, or the hearing, it gets pre-processed. Vision doesn't really get pre-processed. Vision goes right from the, I haven't talked about thalamus yet, but it goes right from the eye to the visual cortex. But when your touch sensors and your hearing get pre-processed a bit. So, but it ends up coming up here. And then layer 3 is considered the output layer. And layer 4 projects to layer 3, and then layer 3 is the output layer. And then the next column over, or hierarchical, so now we're going from like V1 to V2, then this input goes into layer 4 of the next guy. So you see this, and this goes up to layer 3 again, and then it just repeats as you go up the hierarchy. So this is your classic view. The information enters layer 4, gets processed, somehow goes to layer 3, becomes input to layer 4, gets processed to layer 3, gets input to layer 4, and so on. Your classic hierarchy. And of course, this ignores a lot of stuff, like what's everything else doing. And but this is your classic view. We think we understand this pretty well right now. I do want to point out something, that all inference is a matter of mapping many inputs to fewer outputs. It's like if I'm going to label an image, there's millions of images that I might call cat, and millions of images that I might call dog. Or this is true of, you know, millions of different patterns. I might say, that's this song or that's that song. Inference, if this isn't obvious to you, I'll expand on it. But inference is always a many-to-one mapping, and if that's I should mention here too. Mountcastle said that every column in the neocortex is doing the same thing. The corollary to that is that every column does what the neocortex does as a whole. You can't really escape that. If something's going to happen in the neocortex, it's going to happen in every column, because, and that doesn't mean every column processes vision. It means whatever processing elements that are going on are happening everywhere. So that's a background to this. So I'm assuming that anything that happens in the cortex is going to happen in every column. So inference is going to happen in every column, and mapping from many inputs to one output is going to happen in every column. So this tells me right off the bat, there is a many-to-one mapping between layer 4 and layer 3, if I follow that group, right? And many-to-one mapping, the term we use for that is temporal pooling. It means that different patterns over time, they get mapped into a single output. And I can state that even before I understand anything about sensory motor inference. It was just like, that's like a fact. It has to be. If you're gonna put a bunch of patterns and say it's one thing, then it has to occur. And if this is the way I go up the hierarchy, then it has to occur from here to here. So this is and our current theories is that temporal pooling is the term for that. So as your eyes move or your fingers move, this thing is going to be changing, but this is going to be more stable. And it just has to be. There's nowhere else for it to occur. Okay. So so that was that. Then we said, okay, so we could state that. Then we came up with a theory for how the brain could learn sequences and make predictions of high order sequences like melodies and so on. It's a really challenging problem. And we realized we could do that even in a single layer like layer 4. and we use mini-columns. I won't go through that algorithm now unless someone wants me to, but this is the temporal memory algorithm. We said, okay, so I could have a series, if I was listening to a melody, I could have a series of patterns here using these mini-columns, and then I would map all those patterns to layer 3, and that would be the name of the melody. So that's the temporal memory algorithm. And it's very important. There's a lot of really important ideas in how that works. And it's a challenge that people get it at first. So that was great, but it didn't do anything about sensory motor inference. And I avoided talking about, I knew that most of the changes occurred to the brains because we move our bodies. We move our eyes, we move our fingers and so on. That's why most of these changes are occurring. It's not like a melody. Melody happens on its own. But most of the changes coming into our brains because I'm moving my head right now. Everything's moving around rapidly, and these are leading to different changes constantly. Think about the flood of information that's coming in right now as I touch things and look at you. So I didn't understand how that could happen from a sensory motor point of view for a long time, so I just sort of ignored it. And I said, well, we'll figure it out later. Well, we did figure it out. And the answer turns out to be that there's another layer of cells which is representing a location. And that layer cell is and we're going to call that layer 6A. And there is a bidirectional connection between layer 6A and layer 4. This is a very major connection. And what's happening here is that this is keeping track of where the sensor is on the objects in the world. And therefore, it says the location I've sensed this thing. And if I know the location, I can predict what I'm going to sense. And if I know what I sense, it can help me narrow down the location. And so this pairing of sensations or features and locations is the foundation of basic model. It's like that's the foundation of a model. And so this was the idea that neurons in a column could keep track of a location of the sensor. It seemed crazy. How could that be? But we could have deduced it had to happen. That's the only way your finger can predict what it's going to sense. If this thing's getting the input from your tip of your finger, and you can predict what you're going to sense, it needs to know what it's sensing, like what object, and it needs to know where it is on the object. There's no other way around it. And so, and then we realized, oh, it seemed crazy at first, but then we said, oh no, we found these things. We didn't find them. But we know about these other cells in part of the brain called grid cells, which act like a location. So we speculated that there'd be sort of grid cells here. Grid cells exist in the entorhinal cortex, but they were like a reference frame, and they tell you where you are on the object, and of course then you need something else. Well, how does this thing work? Well, we know a lot about grid cells because it's a field of study, and so the basic idea here is that there's a second input coming into each column, a movement input, and as opposed to a sort of feature input or sense thing. It can be a feature, it can be more than, doesn't have to come from a sensory organ, it comes someplace else. So now we have two inputs to a column. One is updating where the location is. Like I know I'm moving in this direction, therefore I can calculate where I will be over time. And grid cells do that. We know that. It's well proven. It's empirical data on grid cells. We speculated it happens in the cortex, but it's known to exist in the entorhinal cortex. So you have a movement information which updates the location, and therefore now you sense something at a new location. And it doesn't matter if I'm going to learn how something feels, it doesn't matter what order I go around it. It doesn't really matter. As long as I cover different locations in any order, it'll keep track of where I am. And then I can build this model. And you need to develop this, if you don't already, this image in your head of a model of like a three-dimensional structure where each location you have some knowledge about. This is what Monty does, about what is there, what was observed there. And so that is the basic of how models are built. Sensory motor models are built in the cortex. And so we guess what these layers mean, but you can see how you're kind of forced to say, well, layer 3 must be doing something like that, and layer 6A, because of these bidirectional connections, must be doing location, and so now you can sort of deduce these things. We might still get it wrong, but I think the actual layers is not as important as the functional role that must be done, but it's a pretty good guess what the layers are at this point in time.
Is there any physiological evidence of layer 3 being more stable as we look at the subject?
No, but I'm not aware of it, but they haven't looked for it because they don't know what to look for. This is a great question. So let's just talk about how a layer of cells represent something. If you look at layer 3, maybe it has 5,000 cells in it, maybe 10,000, I don't know, something like that, right? And it may be, it's very sparse. We know that. That very few neurons are active at any point in time. So you might say, let's say 2% of the cells are active. So maybe you have 100 cells that are active, right? What is stable is that pattern of 100 cells. Any particular cell, by the way, it's only stable if the animal is observing an object that it knows, right? So the animal has to be awake, has to be observing an object that it knows, has to be continually to observe that same object and alert. And then you would expect to see a stability of this 100 cells. Any particular cell will become active 1-2% of the time. So on 1 out of 50 presentations, right? So if you look at the experimental paradigms that people typically do, most of those conditions are not met. The animal's anesthetized, they're showing sinusoidal gratings, they're doing all kinds of stuff where the animal's not awake and alert and looking at something, and they're not, and they'll say, well, the cell, it fired on this image, but it didn't fire on that image. And, you know, and get back to the work that you and Niels are doing. Are these SDRs with these sparse distributed representations, do they have semantic overlap? If they have no semantic overlap, meaning one SDR is randomly chosen from another one, then you won't be able to see a cell assign any meaning to it. One moment it might be a cat, and next minute it might be, you know, a bicycle. I mean, it's just meaningless. It's the step that matters. So it's very difficult to know what to look for. Now they have techniques that they might be able to do this. But again, it's very difficult experiments. Animal has learned an object, recognizes the object, is fixated, moving on that object, is alert, and they're looking at a whole bunch of neurons at the same time. Hard to do.
Right. Pressure might be able to help, yeah.
Right.
But there is evidence that it's more sparse in that layer. Like
Yes.
So
Right.
And has lateral connectivity, which
Yes,
as
right.
I said before, like kind of fits with it.
Right. You've got these 10,000 cells, and what you really want to do is you want the SDR, the sparse distributed, the 100 active cells to sort of self-reinforce. And then another pattern of 100 self-reinforced and so on. The whole sort of mathematics of sparse distributed representations is really interesting. We're not using that at all in Monty today. It's not clear if we have to, but brains do it. All right, other questions? I'm going to fill in a few more pieces here, and then. Is this helpful?
yes.
I'm talking a lot, so I don't know. One thing we didn't understand at first is that there needs to be a concept of orientation. It's insufficient just to know where the patch of the eye is on the object or the fingertip is on the object. You have to know the orientation of the patch. Like I tilt my head left or right, the whole image is on the patch is all rotated. Or if I take my finger and I rotate it like this, my perception isn't changing. I perceive, it doesn't matter if I run my finger this way or this way, I perceive the same object, but it's very different patterns coming on my finger. And so the orientation has to be compensated for in many ways. So there has to be another signal which represents orientation. This is equivalent to head direction cells in the hippocampal complex or elsewhere. So we have an orientation signal, a location and an orientation. And both of those would be updated by movement. Like when I have if the cortex is being told my finger or hand is moving and my finger is moving some way, it could change the orientation, it could change its location. So we have this input coming in here. Both of these can be updated by movement. So now we know the orientation of the sensor to the object, we know the location on the object. Now, this is something we figured out fairly recently. I don't know how many, a year ago, 2 years ago, I don't know. Well, how is orientation taken advantage of here? What we want to do is we want to remove the impact. Imagine if I'm touching an object with my finger, I don't want the orientation to matter. It shouldn't matter what the orientation of my friend. I want to get the same result regardless. Or if the object is rotated, I'm looking at it this way, or I tilt my head, it should be the same thing. I don't want to have, it can't change it. So here's something we figured out fairly recently. It's in the lock down. Something we figured out. I don't have this number somewhere. Okay, so we look at the cortex. We've got columns. There's another part of the brain which is absolutely essential how the cortex works. It's intimately tied to everything in the cortex. That is the thalamus. The thalamus is like it looks like a little bird egg. There's two of them on each side of your head. They're in the center of the brain. And every part of the cortex connects to the thalamus, and every part, and thalamus connects back to every part of the cortex. It's intimately connected. And so, in fact, if you have an eye or finger or whatever, any input to the cortex first goes through the thalamus and then goes up to layer 4, right? And it stops at these things called relay cells. And then the cortex projects back to the thalamus. And so in a major way, lots of details known about these series of connections. But all inputs to the cortex go through relay cells in the thalamus. And if you look at a lot of the literature, they don't always talk about this. They'll say the eye projects to the thalamus, a thing called LGN, which is lateral nucleus. Then it goes to V1, then V1 projects to V2, and V2 projects to V4 in the cortex. Like, this is just how you get into the cortex, and then information flows from region to region, which is true. But then what is also known that every region of the cortex gets input from the thalamus, every region, and every column. So this is where the heterarchy paper comes about, right? There's clearly a hierarchy of projections from V1 to V2 to 4, but it's also in parallel. So, so sometimes these regions are working in parallel, and they're also working hierarchically. And what we now believe is, in the whole hierarchy paper, is that each region is learning objects. Each region or each column in each region is modeling objects. Each one has to have a complete sensory motor interface to the world in some sense. But then these connections are for hierarchical composition, objects composed of objects. So you don't have to, the classic view is that V1 just detects little features, and you have to go bing, 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 and eventually get objects appear. But what we think is going on is everything's recognizing objects. People didn't know it. They didn't know how to observe it. These connections are really saying that the object in V2 has a feature which is an object in V1. So with the classic coffee mug with the logo. The logo would be here, the coffee mug would be here, therefore the logo is part of the coffee mug. But this is recognizing the mug, and this is representing the logo. It's not, and I'm virtually sure not what's happening.
The higher level input, does it also go to layer 4?
What higher layer, what do you mean?
Like, what's going to V2 and V3?
Right, this goes back to the layer 3 projects to layer 4, like layer 3 projects to layer 4, layer 3 projects.
From the thalamus, does it?
Okay. So there's some more details there they didn't tell you. People talk about the main input to a cortical column going to layer 3. Excuse me, layer 4. Layer 4. That's not really completely true. There's another input that goes to lower layers 3, and there's another input that goes to the layer 5, layer 6 border. This could arguably be layer 3, layer 4 border. It's depending on how you look at it. So there's actually 3 different inputs to a column. Most people talk about layer 4 as major input. What I think is going on here is I think this is the motor input, the layer 5/layer 3, layer 6 one. It's basically saying, I'm going to drive these guys to path integrate and update the location. And I think what's going on up here is this is actually related to object behaviors and movement. It's movement related to the object, as opposed to this is movement related to the sensor. The object is moving something on the object. So we can come back to that maybe when we talk about object behaviors. Now, did I answer your question?
So it's the same for V1, V2, and V4. They all get the same composition.
This is basically all look like this, as far as we know. As Murray Sherman has told me several times, he says, everywhere we've looked, we've seen this, but we haven't looked everywhere.
And these inputs from the thalamus to V2 and V4, they come from like higher order thalamic nuclei.
Right.
So the thalamus is like kind of separate, like divided into many different nuclei that get different inputs.
Right. Each region has its own nuclei. Right. And by the way, not only did the sensory input go to it, but the motor input goes to the thalamus too. Or there's a separate there's 2 pathways to the thalamus. They don't call it motors, but we know it's motor. There are these parallel pathways to the thalamus, and from the eye, they're related to the magnocellular and parvocellular pathways from the eye. So there's 2 pathways from the eye, and one represents movement. We're pretty certain about this, and the other represents features. So you have the movement goes to the thalamus and the sensory input goes to the thalamus. What is one of the things, one of the major things the thalamus is doing is part of our theories. They call these relay cells because they look like one spike comes in and one spike comes out. Well, great, what's that for? Not very useful, is it? So they're like, oh, it might be delayed, but these cells are really complicated. They have like 6,000 synapses and very complex architecture. It's a huge amount of complication down here. We can get into if you want to know it. But what's the point of this? If it's a relay cell, what the hell's it doing? You know, that doesn't useful. No one really knows. We now know. We're pretty certain what it is. They're remapping. So what spike is output turns out that it can vary. That these cells, they get more than one input. They have more synapses in different parts of the retina, and so they're like multiplexers. They can basically take an input, remap it, and send it to the cortex. That's the basic idea. There's a lot of subtlety and confusion about how it does that, but there's a lot of suggestions that's what they're doing. And theoretically, it makes sense because what we think is going on is one of the major projections you have going down there is layer 5A, which we think is orientation. And that projects down here, and it says basically, given my current orientation of the sensor to the object, remember this is the orientation of the sensor, like a sensor patch, like your eye, if I tilt my head, the orientation changes. Or if I rotate my finger, the orientation changes. That orientation comes down here and tells this guy how to compensate for it. It says, rotate the input. So, and it has to do that for motor too, because if I'm reading text and I rotate my page like this, which you do all the time, your eyes have to move diagonally to read the text. So your motor behavior changes based on the orientation, and what you sense changes based on the orientation, and this is compensating for it. So now we have the complete system. You've got a system with movement coming in, features coming in. There's a location. Objects are modeled by pairing locations with features. The orientation is used to make sure the movements in the features are rotated properly. We do temporal pooling, and now this is the object coming out. And the reason it goes into layer 4 in this region is saying this feature on this column is the entire object of this column. It's like that. This is the logo. This is the color. I'm going to stop soon.
And you have one column to one column, but just for completeness, like the V2 column will receive L4 input from a whole bunch of other columns.
Not clear, actually.
Okay.
I didn't talk about columns to columns. The way I've always viewed this is we need to understand a single column first, because that was Montel's axiom. And then we have to understand how single columns work together hierarchically, like one column here, one column there. And don't get distracted by all those other columns. But now the question is, is there convergence onto V2 or something like that? I don't actually know the answer to that question. There's evidence that there is, but that convergence, I'm hesitant because I think it can be interpreted wrong.
Isn't like it's often physiological, as in like
right.
If you measure the response in V2, it's like, oh, this can see a lot, but it's not necessarily the case that that's because of convergence of input. It could be more
right.
like
imagine you have a visual column and you're looking just like through a straw, it's like one column, and it sees a cat, right? And you can't only recognize a cat by moving the straw around a little bit. Okay, I see there's a cat. As Niels is saying, the output will be cat, and this guy will be getting input for cat, and that looks like a broader area of the cortex, right? Because it's everything that this guy could see as moves around. So physiologically, as Neil said, this would be broader area, would respond to anywhere in the cat. And if you look at this layer, it's only going to respond to one part of the cat. But if they knew how to look at this layer, they would see it was responding to cat. But as we said earlier, they don't really know how to look at this layer because it's sparse and there's all these things. And so, so these are testable hypotheses, but it has to be like this in some sense. It's deduction. It just has to be. People get annoyed when I say that, but that's the way I do it.
The surface area is smaller though as you go up. So like either the columns are getting smaller in V2 and so on, or they're getting conversion input.
Well, okay. Not necessarily either one of those will have to be true. first of all, interesting, going from V1 to V2, V2 is actually a little bit larger than V1. So you have you typically, you see the same with S1 and S2, the first two regions are really large.
Yeah.
And then they start getting smaller quickly.
Right.
I interpret that as most vision actually occurs in V1 and V2. And you only need 2 layers to learn a compositional structure. And so there's a huge amount of memory in V1 and V2. It's going to be learning a lot of the world. And then these things get narrower very quickly, and they become more multimodal and other things. So that's the number one thing. It's not a strict convergence going up. And there are multiple ways that a region can get smaller with or without. I guess I want to avoid the word convergence because that's essentially saying, oh, this column can only understand something if it's looking at a larger part of the world. It can be physiological. It doesn't. I haven't worked it all. I don't like to. If you really want to, we can spend some time on this. But I don't think that's the right way to look at it. It's not the right way to look at it. Isn't like I have to have convergence of columns because I think the whole system can be built with a single module, single column on top of a single column on top of a single column. It won't be the most efficient system, but it would work. So you don't have to have convergence of columns for this to work. We can build our first compositional object with 2 learning modules if we want to do that.
Yeah, I guess maybe one way of saying it is like, it's not an issue if there is convergence.
That's right.
It can be accommodated.
Right.
It's not necessary.
Right, that's a good way of putting it. Don't fool yourself and think we have to have it. We don't. It can exist, but we don't have to have it. In fact, the theory we came up with compositionality is the following. You know, you have, imagine this is V2, and you have a bunch of columns here. And here's V1, roughly the same size. You know, the columns can be a little different, but roughly the same size in area. Maybe V2 is a little bit larger, like I said. The theory that we worked out, and this is the last time, it was the first kind of pieces together when Vivian was here back, whenever it was, we were working on this problem, is to understand that the way compositionality works is it's on a location by location basis. So imagine this is getting input from some part of the retina. Let's see here. Here's the eyeball. The retina is back here, right? Imagine we're getting input from this little patch right here, okay? This column is getting input from that patch. And this column is also getting input from a patch that is co-located with it. They're essentially pointing at the same point in space, right? Like V1 is looking at the retina, it gets input from the retina, V2 gets input from the retina, and this column, these columns are both centered on the same part of the retina. When we learn compositional structure, and this is all in the hierarchy paper, we're essentially saying this co-alignment is essential. You learn what the system is doing. If I have a coffee cup with the logo on it, that's supposed to be Ellen. It's basically saying at this point on the logo is also a point on the cup. And at that point in physical space, I can assign this point on the logo to this point on the cup. It's on a location by location basis. So these 2 guys, as you move around, it says, oh, I'm gonna learn to connect. Basically, this is a sort of a bidirectional connection here. I'm gonna learn at this point of the cup, I'm at this point of the logo. And I'm at this point of the cup, I'm at this point of the logo, and so on. And this allows the logo to be morphed and change and rotate and all kinds of weird stuff, and still learns it. So when we build compositional structures, we can really do this with just 2 columns. That is sufficient to do it. Not the most. It's sufficient, not efficient. The MS of the theory tells us.
And yeah, and that location by location also can store the orientation, which is why
Right.
like it can depend and stuff. But maybe it's worth
It's
checking that makes sense because that'll be important for the location by location for the behavior.
It has to know the idea of the object, like this is a logo in the cup. It has to know the orientation of the object, like it's rotated here. And it needs to know the scale of the object because it could be bigger and smaller. We learn this stuff really quickly and instantly. So we learn these things for every, and the surprising thing was it has to be done on a location by basis. But if you do that, then the logo can have distortions and be twisted and can do various things. So these are the 3 things we have to learn as part of the model. We have to say the feature coming into this guy is rotated and at a certain scale at this location. And we'll go through the circuitry for how this actually works. It's in the paper, but you can see how this actually works.
And then maybe it's worth
Go ahead.
highlighting that I don't know if I'm trying, but just that like that informs inference. So like if you learn like a particular mug with like a funny logo or whatever, that information coming in about like what the pose at a particular location of the logo is can tell you, oh, that was that mug that had the weird thing. But also it can assist prediction in that, let's say, because of the other side you saw the mug, this is that weird mug with the weird logo. You can predict at a location a particular orientation scale of the logo. So yeah, I guess it's just that the location by location base is important both for the feed forward and for the feedback,
Right,
which comes into, I guess, the anatomy.
right.
And I'm just emphasizing it because when we come to talking about the behaviors, and that's location by location,
Yeah.
then it's helpful to understand that. And
I think when we
yeah.
talk about that, we should project the images from the paper because they're nicer
Right.
than these paper images.
So are we saying that input to V2 is more receptive field, or is it the same receptive field as the input to V1?
It could be typically it's a larger receptive field, like from the retina itself, more ganglion cells in the retina converge onto V2. But that's not important.
Lower resolution.
It gives a lower out, right? It may be very, very efficient and useful, but it's not theoretically essential.
And yeah, and it would fit with like this learning like a coarser model and this learning a more fine-grained model. So like there's only so much information that any cortical column can store. So if it's getting small inputs or like kind of fine detail inputs, realistically, it's gonna learn kind of a fairly small but like detailed model. This one, it's getting over a larger surface area, so it cannot necessarily distinguish details as much, but it can build like a coarser.
I always thought this part was leave it just to the hierarchy and not like I make the template pool in the hierarchy take care of that part, because I can't, like, how big can this receptive field go, right? Because we
They get bigger, V1, they're really small.
yeah,
right.
but like if we go to V2 and V4, can it cover like the whole object?
Well, it's not clear, it's not, I mean, V2 definitely gets input from the retina. I don't know about V4.
Right, so it cannot be that big that it can cover like larger objects like
Well, it would be bigger than V1. I mean, I think there's a lot of evidence that the convergence from the retina onto the cell in V1 and versus cell in V2, there's more convergence onto V2, makes the broader area. You see, that's also property in the somatosensory cortex. So that's an empirical observation in some sense. We accept it. I think as there's a couple of advantages that Niels mentioned. One, it's like, the V1 is the highest acuity you can get. There's nothing more refined than that. So if I'm reading the smallest print that I could possibly read and recognize, it's almost certainly happening in V1. If I'm saying that's the smallest, and V2 would be too blurry, right? But if I have a very big letter, V1 is too small because you would have to like integrate over a huge amount of area, and it's just not very useful. But V2 could do it in a more reasonable way. So that makes sense from a just sort of scale perspective. It also makes sense that when you have composite objects, it's really hard to come up with an example where a child is bigger than the parent, right? It's almost always the child object is smaller than the parent object. So that also makes sense. In fact, if it was bigger, then you would probably flip around and say one was. But from a theory point of view, it's not essential. It's just that they have to be two different objects and they have to have the ability to have the same, at times, the same point on them.
so they're looking at the same point, but one is focused on the
Right, one is attending to the logo and one, so when you see a new object, I picked that one up, it's got water in it. I don't know. Well, that's not a great one, but it doesn't have much of a logo. Oh, it says Kirkland on it, right? When you see a new object, you say, oh, that's a bottle. And then you tend to focus on the details. And then you start saying, oh, it's Kirkland, it says this, it's that, oh, it's got this little funny bottom, there's a cap. While you're doing that, I believe what's happening is there's a higher region that's still saying this is the bottle. And you're attending to all these components and in series building up the model of the bottle. So now I say, oh yeah, I see there's the Kirkland logo and there's this thing and there's that thing and there's this thing and I can look on the back, oh, there's this thing. And now I have a model of this object because I knew it was a bottle and I'm looking at the intended features one at a time. So that part of our mechanism for doing compositional structure is to keep, we were talking about this earlier about your unsupervised learning. One region or one column has to be maintaining, I'm looking at this bottle. And the other one is saying, I'm looking at the features on it individually and recognizing them individually.
But how does the higher level know it's a bottle without the lower level?
Because the high level also has, it basically has its own feature detections that comes straight from the retina in some sense, right? The boundaries of the bottle can be learned. And these are grosser features. And then only when you basically attend to a subset does the bottle stay the same. I mean, I don't know how else it could work. I mean, it's sort of like you deduce it has to do this. I can't, maybe I just failing in my brain, but it seems like this absolutely has to happen. Once you understand it, you're like, there's no other way of doing this. You have to be able to assign these individual components to a larger object, and you do that by moving around them, looking at them one at a time.